drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. My pleasure. Can you start off by just telling everyone a little bit about who you are and what your lab does?
Yeah, I'm a professor in the Department of Psychiatry here at Stanford. And my lab has been studying the neuronal underpinnings of sleep and wakefulness for over 30 years.
And sleep and wakefulness are really interesting because in many ways, these are mysterious phenomena, but at the same time, we're all very familiar with these things at a personal level. You know, we all go to sleep and we're all awake. We all know what it feels like to be in a state of high vigilance, in a state of deep sleep, and so forth. At a very high level, when neuroscientists talk about behavioral states and brain states, what do those terms mean and how do you start to think about them?
Yeah, that's a great question. So, we start from the basis that all animals sleep. So all animals have cycles of activity and rest. And we define sleep is essentially a behavioral state that has one particular property, which is sleep rebound or sleep homeostasis. That means, as you just mentioned, that when we are awake for a longer period of time, then we feel this urge to go back to sleep, and this sort of sleep drive gets stronger, the longer we've been awake. So that's different from just the circadian cycle, where we just feel that we just want to go to sleep because it's nighttime. So the neural processes that govern this sleep drive are essentially what we're studying when we're studying sleep. And the manifestation of this drive is in changes in the activity of in mammals cortical neurons. And we monitor those changes using an electroencephalograph, which can very broadly measures cortical activity. So sleep and wakefulness can be defined objectively with an electroencephalograph by looking at the shape of the waves, the waveforms, essentially in the electroencephalogram.
So there's behavioral changes that correlate with these different patterns of brain activity. So obviously, when animals are sleeping, they're not moving around as much. So there's macroscopic things you can see that you can see in terms of an animal's external behavior, and those are mirrored by just general patterns of global brain activity that you can measure with EEG and things like that.
That's correct. And of course, the EEG can be measured only in a few species, in lower vertebrates, and actually in insects and other animals. It gets obviously very difficult or impossible to get an EEG recording. So then we measure sleep strictly behaviorally, and we look for periods of quiescence that again have this property of sleep homeostasis. That means that when we interfere with the with sleep, we extend the wake periods, then we see this increased drive to fall asleep.
Mm-hmm. And so with that kind of homeostatic regulation, so if you deprive an animal of sleep, you prevent it from falling asleep for longer and longer periods of time, a sleep drive will build up, and then it will sleep more to make up for that. That would imply, right, that there's some essential function being served here by sleep.
That's correct, and we're still trying to figure that out. There are several hypotheses out there as to what the main function of sleep is. Some researchers argue that it's synaptic scaling. That means that that there is an increased synaptic or communication between neurons. That's what synapses are. So there's increased communication in the brain that needs to be reset every day so that we don't go just wild in terms of neuronal activity. So that's the synaptic downscaling hypothesis by Giulio Tononi and Chiara Scioli, essentially. And then there's another, well, there are actually several hypotheses. Another hypothesis is that sleep is necessary to clean up your brain, so that during sleep there's this increased flow of in the brain that allows to clear metabolites and toxic substances that accumulate during wakefulness. But there are many others. There are other hypotheses that point to, for instance, DNA repair as one of the main drivers for sleep, and also metabolic recovery. There are a bunch of hypotheses, none of which have been proven yet, or we only have partial pieces of evidence that demonstrate that there is that sleep is essential for some of these functions.
Mm-hmm. And so just being agnostic about the evolutionary purpose of sleep, when you look at brain activity with EEG in the mammalian brain, where you can measure that activity, what are the major phases of sleep, and what do they look like in terms of general levels of brain activity?
I'm not sure if I understand that question. So
Yeah, so if you compare sleep to wakefulness and you compare, say, non-REM to REM sleep,
Mhm.
these are different behavioral states and they have very different, there's very different patterns of brain activity you can observe through
Yeah.
EEG.
Mhm.
What do those actually look like for the different phases of sleep in comparison to wakefulness?
Oh, I see. So, yeah, I mean, again, that's based on the EEG and how we know or what we know about the EEG. So, during wakefulness, there is this um, very complex pattern of activity throughout the cortex, which is what we are measuring in the EEG. Um, and the EEG can be deconstructed into different waves or waveforms. We call the alpha, beta, and so forth, and delta. So yeah, during wakefulness, there's a predominance of alpha and gamma and high frequency activity. Okay, and that is in contrast to non-REM sleep, which is characterized by a very high contribution of what we call the delta waves, which are slow wave activity, and that is essentially reflects synchronized activity throughout the cortex. That means that many neurons are firing in synchrony during a non-REM sleep. And in contrast to that, REM sleep is characterized by a very high predominance of what we call the theta activity, which is a activity at the 7 to 11 hertz range, and that is also present during excuse me during wakefulness, but again REM sleep is very prominent on that in that frequency, and of course REM sleep is also characterized by almost complete muscle atonia. That means that all muscles except for the eyes and the breathing muscles involved in breathing are essentially uh, lack activity. So yeah, those are the broad picture, the differences between the three vigilant states.
Mhm. So there's these very different patterns of brain activity that characterize each of these states. What is like the neural basis for those brain state changes? Is it just that different neuromodulators are either, being used a lot versus very little? What's actually accounting for, do we know what accounts for the very different brain states that you can measure with EEG?
Yeah, well, that's we're still trying to figure this out. I mean, it's a very complex um, question, if you want to, if you allow me to say that. So we don't know. We don't know. What drives those complex patterns. We cannot really predict them very well. Um, in general, and again, that's a very broad picture. There are a bunch of excitatory transmitters and modulators that are more active during wakefulness than during sleep. But we see all sorts of patterns both during wakefulness and during sleep. And in fact, there's this phenomenon that was characterized about 10 years ago by Vlad Vysotsky, which is called local sleep. So there is this uh, sleep state within wakefulness, and also vice versa, like a uh, wake state within sleep that that you can detect in different modules, quote unquote, in the cortex. So there is not a simple answer to what is turned on during sleep, what is turned off during wakefulness. It's just very complex.
Hmm. So there's both external and internal variables that influence when animals sleep. So for example, an obvious environmental variable would just be where you are in the day. So for a diurnal animal like humans, we're generally awake when the sun is up, and then we're generally asleep when the sun is down. And we all know that this has some influence on our sleep patterns. We tend to get more sleepy when the sun is down, and we tend to be more awake when the sun is up. But of course, there's internal factors as well. And if you sleep deprive an animal for long enough, you stay awake for long enough, eventually you'll get so tired that you'll even fall asleep in the middle of the day. And so, what are some of these internal factors that build up that drive sleep? Is there something going on with metabolism where certain molecules are building up and serving as sleep drivers? Is it sort of have to do with, say, the brain using up neurotransmitters or running out of fuel in some sense? What are some of the major internal drivers of sleep?
Yeah, no, that's a fantastic question. So yes, so we're still trying to figure out what those variables are. But we know that there are a bunch of them, and that some of them are running in conflict at many times during the day and during the night. So for instance, you actually mentioned already quite a few of them. So the circadian rhythm, of course, is a main driver. Metabolism is a main driver. Of course, the brain uses 20% of the energy of the whole body. So it's important that the energy allocation to the brain gets conserved, and of course, sleep helps with that. So what else? There's actually a bunch of variables. Emotions, and I think we can expand onto this. We all know that very strong positive emotions of excitement, and in the wild that would be access to, for instance, a high density of food or resources, or the availability of a mating partner. So that stimulus obviously inhibits sleep, because it's a very good opportunity to engage in those survival or mating opportunities. So then that inhibits sleep. Negative emotions, and in the wild, the most negative emotion is to have the risk of survival. So if you have a predator behind you, obviously it's not a good idea to fall asleep. So a predator behind you engages the fight or flight response, the stress systems, and that very, very powerfully inhibit sleep. So those variables are integrated in several circuits essentially in the hypothalamus, and then those conflicts are computed, are sorted out, and then the decision to wake up or sleep is being made by that bunch of neurons, and then that information is conveyed to several transmitter systems, including the monoamines, norepinephrine, serotonin, dopamine, and others, and that essentially would wake you up or inhibit activity to allow for a sleep path.
Mm-hmm. And so with sleep deprivation, so we've all had the experience where, we stay up longer than we should, or that we would like to. And, gradually you have this buildup of a sleep drive. We get more and more tired the longer we've gone without sleep. Is there anything that accumulates over, with that slow time course in the body that is at least partially responsible for that gradual buildup of sleep drive?
Yeah, again, a very interesting question. So if you asked that question, 20 years ago, the vast majority of neuroscientists would have answered adenosine. Adenosine is the main molecule that accumulates during wakefulness and is released during sleep. And we all know, of course, that caffeine is an adenosine inhibitor, and that's why one of the mechanisms by which we can stay awake for longer. But research in the past decade or so has actually tempered that hypothesis, and it's less. The evidence is not so strong. Let's put it this way: that adenosine is a main player. There's a lot of work in the metabolic neurometabolism field and also in the sleep field, trying to figure out which substances, which metabolites could serve as markers of sleep drive. And the truth is that it's unlikely that a single parameter will drive sleep need. So it's probably a vector, combination, multidimensional construct that drives sleep. And there are, in addition to the neurometabolic parameters, there are also studies that indicate that there is a shift in the sensitivity of a very small group of neurons that again changes its sensitivity to neuromodulators, and that is essentially what drives sleep pressure. But again, evidence supporting this is only strong in invertebrates, in Drosophila, in the fruit fly, and really not so much in other organisms. So we still have to be cautious about how to extrapolate those findings.
Yeah. So there's many variables that interact or compete with each other in some sense that get integrated together and determine whether an animal falls asleep or wakes up. And this is a very complex phenomenon. The brain has to keep track of whether or not you're in a rich social environment, whether or not you're safe or there might be predators around, what time of day it is, how long it's been since you last slept, all of these many factors. But you said a decision gets made in the hypothalamus, essentially, which is a deep structure in the brain. And I'm interested in that term, like a decision gets made. It's an interesting way of putting it. And one of the things that's kind of interesting is, despite the fact that wakefulness can change on a slow time course, and we can sort of gradually get tired the longer and longer it's been since we sleep. Ultimately, the decision or the switch in the brain happens quite abruptly. We fall asleep in a matter of seconds. How does that sort of integration happen within the hypothalamus, and why is it that this sort of very sudden transition ultimately happens, where we fall asleep within a matter of just seconds?
Well, it's really our perception that we fall asleep in a matter of seconds. The brain takes longer to fall asleep, actually. But we could also explain that that the speed by which we fall asleep in terms of cortical dynamics. So there's essentially what we call they're emergent properties of very complex circuits. And at one point, when you synchronize enough number of modules of the cortex, then you essentially have to fall asleep. That's in very broad terms, that's how we would explain this fast dynamic. But yeah, indeed, the decision is being made in the hypothalamus because that's what that's what lots of neurons do. They integrate, they compute their inputs, and then based on those inputs, and of course that's an online decision, then the neuron fires or it doesn't fire, and that's a decision that has consequences downstream. So that activates or inhibits or doesn't activate certain circuits, and that's essentially how it works.
Mm-hmm. And so, is there a particular circuit or subregion of the hypothalamus where this decision is made ultimately?
Yeah, we've learned that's actually work that we've been conducting for the last 20-something years, almost 30 years. So we identified this group of neurons in the lateral hypothalamus that we call the Hypocretin neurons. So other researchers call them Orexins. And they're all localized in the lateral hypothalamus. There are about 5,000 neurons in rodents, about 50,000 neurons in humans that are responsible for making that decision. And how do we know this? Essentially because when we don't have those neurons, we have narcolepsy. And narcolepsy is a sleep disorder that is characterized by disrupted sleep-wake patterns. Essentially, narcoleptic patients that lack hypocretin neurons, they cannot, their brain cannot make that decision appropriately. So they don't consolidate sleep. They have serious issues in state boundary control.
And so these hypocretin neurons, they're in a particular subregion of the hypothalamus. They're important for regulating sleep cycles. If you don't have those, you have narcolepsy, so you have very disordered sleep-wake regulation. What is hypocretin or orexin? What type of molecule is it? What does it do at the cellular level?
Yeah, well, the hypocretins are orexins are two peptides. They're produced by the same precursor. And peptide transmitters are very widespread in the brain and also in evolution. They're molecules essentially that evolved to connect circuits at a slow pace, so to speak, in a slower time scale than fast transmitters. And they work by binding to receptors in the postsynaptic, what we call the postsynaptic neuron, the recipient neuron, and they, that the binding of these neuropeptide to the receptor triggers a whole bunch of intracellular events that result in either excitatory or inhibitory action on the postsynaptic neuron.
Mm-hmm. So these neurons in the lateral hypothalamus, these hypocretin neurons, they are, in a sense, maybe this is simplifying things a bit, but they're the decision makers. They're really important for dictating whether or not the animal will fall asleep or wake up. But with certain exceptions, animals generally, the entire animal falls asleep, and the entire brain more or less falls asleep, unless there's exceptions there that we might talk about. How does that decision in one region sort of get broadcast to the entire brain?
Well, that's because, again, the hypothalamus and that particular region of the hypothalamus where the hypocretin neurons reside, this region connects to very broadly throughout the brain, and in particular to nodes that are really, really, really important to activate the whole cortex very quickly. And those are what I was referring to before as the monoamines, norepinephrine, serotonin, dopamine, histamine, and in particular, norepinephrine that we also, described a while ago as being really a very, very powerful, wake-promoting transmitter, is very, very tightly connected with hypocretin. So a signal from hypocretin would activate norepinephrine very, very efficiently, and that would essentially light up the brain and wake you up. In very simple terms.
I see. So there's these neuromodulators in the brain. They the cell bodies where those neuromodulators are made, they are in certain parts of the brain, but they send projections throughout large chunks of the cerebral cortex and much of the brain. So these hypocretins can broadcast the signal widely by tapping into those neuromodulatory cells, which are then gonna sort of communicate broadly across the brain all at once. That's
exactly what happens. That's a very, very nice way of describing it. Yes.
And so you mentioned norepinephrine, which is important here. So I think another name for norepinephrine is noradrenaline. So people might naturally think that's a wake-promoting neuromodulator. Is that true? And if so, how exactly does a neuromodulator like that one promote wakefulness? Is there something sort of special it does to the neurons, or something different that it does in comparison to other neuromodulators?
Yeah, well, norepinephrine is indeed a very special transmitter for several reasons, but one being that a lot of the neurons in the cortex do express these receptors for norepinephrine. So it's a signal that very efficiently reaches to very, very large areas of the cortex and very efficiently stimulates their activity. And when I'm saying efficiently, it's because at any given time there's a very tightly regulated balance of excitation and inhibition in the cortex, and norepinephrine can, again, in very simple terms, override that balance so that wakefulness predominates over pretty much anything else that is going on in the cortex at that time.
Mm-hmm. Interesting. And so what are some, so I would imagine, so when you're awake, norepinephrine levels are generally quite high, and when you're asleep, I would imagine they're quite low. What is sort of the mix of the major neuromodulators when you're awake versus when you're asleep, say in non-REM versus REM sleep? Those must have to change in different ways to define each of those phases.
Yeah, well, I wish we knew, because we can only until now. Now we're starting to have the tools to answer that very question, and that is a very, very very important question, actually. What's the mix of neuromodulators and transmitters at any given time? Norepinephrine, for instance, the activity of norepinephrine neurons varies actually relatively little during wakefulness. So if we monitor the frequency of activity of norepinephrine neurons during wakefulness, it varies between 3 to 5 hertz. So not a whole lot of variation. And during sleep, then it falls to 0.5 to 0.2 hertz to 1 hertz. Okay. And during REM sleep is essentially 0.1 or 0 hertz. Okay, so that's the variation. And on top of that, of course, we have a whole bunch of other transmitters. And dopamine, for instance, has a tonic activity, around again 5 hertz also. But one of the characteristics, one of the features of the dopamine is that it has this phasic activity, so just bursting activity essentially, that is responsive to stimuli or prediction of prediction errors, prediction of errors, as we call them. So so there's more complexity added onto that 3 to 5 hertz tonic activity in dopamine. Serotonin is also has very variable, a very broad dynamic range during wakefulness. And again, we don't know exactly how it modulates transitions to sleep, and what is necessary, what is permissive for sleep. But just to go back to your question, so we only know, again, single channel, so to speak, recordings. So recordings of a single transmitter at a time. And now we have sensors that have been developed recently by a few of our colleagues that will allow us to essentially have a better description with a multi-dimensional vector that allows us to really understand which, what the contribution of each single transmitter to the brain states.
Mm-hmm. And so I want to talk a little bit about sleep architecture. So we fall asleep, but sleep itself is not just one state. There's different phases of sleep that have very different characteristics. So there's non-REM sleep. There's different phases of non-REM sleep, including deep sleep. There's REM sleep, and we've talked a little bit about those already. But where does this architecture arise from, and what's the significance of transitioning into non-REM and then REM and then non-REM and REM sleep throughout a period of sleeping?
That is a very difficult question, and I don't think we know the answer. So for people interested in memory consolidation as one of the functions of sleep, they could argue, well, we need to cycle through those phases because that's essentially that's essentially how the way that memory circuits work. Okay. I would argue that that's actually not a very solid argument, because there animals that have very different sleep architectures with very different brain configurations, and we can talk about, also very special cases of sleep. And but anyway, so we really don't know why there are these phases of sleep. And if we compare, for instance, rodents and humans, we essentially go through very similar phases, but of course humans have consolidated sleep. So we sleep for over 8 hours, and we have several cycles during these 8 hours. Rodents don't have the consolidated sleep, so they wake up a lot during their day, which they're nocturnal animals, so they sleep during the day. And their cycles are much shorter, and their transitions are much faster. So why do we need those cycles? Why do we need the sequence of those of the phases of the substates between sleep? We don't really know very well.
And so you mentioned that there's obviously a lot of variety in nature in terms of what sleep architecture looks like. Some animals sleep much more than others. Some of them, their sleep is more fragmented. They wake up more times, and their sleep is broken up more. Are there any animals that have sort of bizarre or deviant sleep patterns that you think tell us something important about some of these different phases of sleep and what it might be doing? Animals that either, spend a lot of time in one phase or another phase and just have very different sleep architecture.
Yeah, I think, well, that is also a fascinating topic in sleep, of course. The ratio of REM sleep versus compared to non-REM sleep, it varies dramatically across species. We have I think the extreme is the bats, which are only awake for a few hours, essentially at dusk, which is when the insects are available, their food is available, and then they fall asleep for the rest of the day for 20 hours. And most of this time is REM sleep. Again, we don't know why. I mean, it really doesn't fit with memory consolidation that you need REM sleep for memory consolidation. I mean, we don't know why these animals have so much REM. There is also some correlation between metabolism and REM sleep, and that was brought up by Jerry Siegel. So ruminants that have slower metabolism have more REM sleep. Marine mammals don't have much of REM sleep at all. And also one very peculiar aspect of sleep in these marine mammals is that they sleep with half of the brain, so while the other half is active. So that's what we call the unihemispheric sleep. And also very recently, this example of the penguins. So there was a group that recorded penguins in the wild, and they showed that while they're incubating eggs, which is a period of their life when they are very exposed to predators, so they have to be very vigilant, and they sleep at in 2 to 3 second intervals. So it's essentially they are, but they have these sleep episodes like 10,000 times a day. So overall they sleep 12 hours, but they sleep only in very, very short sleep bouts.
Wow. So when they're incubating the eggs, they have to stay on the eggs to keep them warm, especially in that cold environment. Obviously, if they can't move, they're really vulnerable to predators and things. So you're saying they essentially just engage in many, many small little bouts of micro sleep throughout the day.
That's correct. That's right. Well, and the same animals, when they are at sea, they have much more relaxed sleep. They can sleep well, and they have much completely different sleep architecture. And also, there are other examples of extreme sleep, like the flycat birds, for instance, that when they migrate, they are essentially sleepless for a very, very long, 2-week migration period, right? And also the big changes in their metabolism as they migrate, and there are some really incredible adaptive strategies for those conditions.
So there's a lot of diversity in nature with sleep architecture. And it's really complicated. So it's going to relate to a lot of things. Obviously, the lifestyle of the animal, when its food's available. You mentioned, bats, their food is only available one time of day. So they're sort of set up to be awake at that time. They don't need to be awake. At other times, they can also hang upside down from the cave. So they're pretty safe and they can afford to do that, in contrast to the penguins who have to be prepared to make a run for it or whatever when they're staying awake to incubate their eggs. So there's obviously all these ecological variables that constrain and dictate what sleep architecture actually looks like in a given species. But within a single species, so an experimental species like a mouse, say, have people been able to manipulate sleep architecture in ways that allow us to understand the contribution of individual parts of sleep to whatever it is that sleep is doing? So, for example, can you selectively inhibit REM sleep but not non-REM sleep, or vice versa, and see what kind of effects that has on memory consolidation or something like this?
Yeah, that has been done, and beautiful experiments in that direction have shown that by inhibiting REM sleep, and not only REM sleep but actually yes, the theta oscillation that I was referring to. So that is driven by one brain area in particular during REM sleep, is essential for memory consolidation. So that is, those are beautiful experiments. The problem is that it's very hard or impossible to disentangle one part of sleep without affecting the other. So if you affect REM sleep, you're going to necessarily affect non-REM sleep.
I see.
So I would say that the evidence, again, supporting that there is something going on during REM sleep for memory consolidation is quite solid. But we were also quite excited, a few years ago when this story about memory replay during sleep came about. So that essentially sort of to introduce this concept. Essentially, researchers were recording the activity of neurons in the hippocampus as an animal was exploring a maze, and then there are these neurons that are called place cells that fire whenever this animal encounters a particular space in that maze. So you can actually predict the position of the animal in the maze based on the activity of those neurons. It's quite amazing. So there's this group at MIT, Mike Wilson, who showed that during sleep there was the sequence of activity that had occurred during wakefulness as the animal was exploring the maze was replayed during sleep. So that provided a very attractive hypothesis that the animal is replaying so that it remembers where it has been the day before, and it potentiates the synapses, blah 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 blah. So there are a whole bunch of theories came from that observation, but then later studies show that there's replay also during wakefulness. So it's not sleep is not necessary for that replay. So what is sleep for then? That's that's a question we're trying to address. And going back again to your question. So is REM sleep more important than non-REM? It's still not clear.
I see.
If you manipulate one aspect of sleep, because everything is connected, it's very likely that you're going to affect others, and then you're going to, the conclusions are going to be, much harder to.
So if you put a mouse through a maze, it will explore the maze. It will take some trajectory through the maze, and you can essentially see the trajectory the animal is taking by just looking and recording from specific neurons in the brain, and the pattern with which they fire tells you the trajectory the animal took through physical space. And you're saying that during sleep, but also when they're awake and they're just in a state of quiet waking, not really doing anything, the same patterns that fired when they were actually going through the maze will replay in certain circuits of the brain while the animal is disengaged from navigating the maze, either while it's awake or while it's asleep.
Mm-hmm. That's exactly right. Yeah.
And so I would imagine the natural interpretation of this when you see this replay during sleep is, it naturally makes you think about dreaming. Like, oh, okay, I often dream about things that happened to me recently. Maybe I dream about important things or emotionally salient things more. And so maybe, the mouse, if we speculate, is dreaming about what happened to it. And that essentially would correspond in this interpretation to the brain replaying certain patterns of activity for the purpose of memory consolidation. Is that the basic idea? And if so, it sounds like maybe people actually demonstrated that's really what's going on?
Well, in regards to dreaming, no, they're actually separate. Dreaming is a different rearrangement of activity that occurs usually during REM sleep, but also during a deep non-REM sleep. So the replay that these experiments are referring to, essentially, it was a snapshot of the activity of neurons during sleep, and it was surprising that indeed was almost identical to what the animal had experienced the previous day. So. I also became very excited about this finding, honestly. But again, one has to put it in the context of, if we only record one channel of activity and only those cells, the place cells, we're missing really the big picture of what is going on during sleep, and that is, again, that is what we're trying to get at, with the current technologies. Yes.
When that replay activity is observed during sleep, what phase of sleep is it in? Is it a non-REM sleep or REM?
It's non-REM. Yeah, it's non-REM.
I see. So that would be if REM sleep is most closely associated with dreaming, then, so presumably the rodents were not dreaming in non-REM sleep.
That's correct. Yeah.
Interesting. And so one of the things I want to talk about as well is just like overall sleep depth and sleep quality. So taking those in two pieces, when we talk about sleep depth, I would imagine that the coarse-grained way you measure that is just you try and poke the animal or wake it up, and the harder it is to wake it up, the deeper its sleep is. And obviously, again, we've all had experiences like this where sometimes you try to wake someone up and they wake up very easily. Sometimes it takes much more effort to wake someone up. What are the deepest phases of sleep, and do we know anything about what's causing it to be deep?
Well, the sleep depth is defined by the amplitude of the slow wave activity, essentially the delta waves I was referring to at the beginning. So, and that correlates, as you mentioned, with the arousal threshold. So, the higher the amplitude of those delta waves, the more difficult it is to wake up an animal or an individual. So that is, again, in general terms, but it's not always the case. And also, there is a very interesting, I would say, disconnect between what we perceive as a good night's sleep, or a sleep where we have slept very deeply, and again, our perception of sleep and the actual, EEG recordings. They sometimes, most often they do connect well, but they do overlap well, but in some instances they don't. And so it so happens that there's actually a bunch of people who complain about insomnia. They go to their primary doctor. The primary doctor refers them to the sleep clinic or sleep specialist. So they do a sleep study, and it turns out that many of these people don't have they have perfectly normal sleep, but they do complain about not sleeping well.
Hmm.
And it's just not an anecdote. It's really a lot of people, have these this disconnect. So what is it so what happens then? Is that the case that we're missing something in the EEG, in the electrical recordings that do not really reflect what is going on really in the brain? Is it that we are missing some features of the EEG in the normal protocols of analysis? So we still don't know, but it looks like sleep in these patients seems not to be actually perfectly fine. So there are some parameters that are different from in these patients from the healthy controls.
Yeah.
Yeah.
So if you measure sleep-wake cycles with EEG, EEG is really good at defining when you're asleep, when you're awake, non-REM versus REM sleep, but it's a pretty coarse-grained measurement. And it sounds like you're basically saying there's many examples of people who their EEG looks perfectly normal. They're going through their sleep-wake cycles like people who are well rested do, and yet they say that they're not well rested. So the natural thing to think there is that the EEG's just not able to tell us the full story. There's something going on that we can't see.
That's exactly right. Yeah, that's with the experimental animals, of course, we have the ability to go deeper at the EEG analysis. We can add more electrodes. We can really have a better idea of what is going on in the brain. We can actually, be invasive and then insert electrodes in the brains of the animals and really see if there's, what else the EEG is not telling us. And but of course we cannot ask the animal whether they feel tired or not, and if they had a good night or not. So that's essentially we're trying to come up with additional measures in the EEG that will be able to tell us in humans whether this a person has a healthy sleep or not.
Mm-hmm. And something that's very obvious from experience is that stress affects sleep quality and sleep duration. And, when people are really stressed out, they generally report having trouble sleeping or they wake up frequently, things like this. Cortisol's the major stress hormone that you usually hear about as a marker for overall stress levels. How does cortisol change throughout a normal sleep-wake cycle for a human? And what is the role of stress and cortisol in sleep?
Yeah, well, cortisol has a circadian cycle, right? And so it peaks during the early hours in the morning. And what does it tell us? So those are again similar to what I told you with the dopamine tone. So cortisol has its day-night oscillation, regular oscillation, but when we have a stressor, then that oscillation gets disrupted. So the presence of a stressor increases dramatically the release of cortisol, and that affects the whole body. It puts the individual in a state of either ready to fight or ready to flee. And that, of course, affects the immune system, affects just arousal levels, affects the cardiovascular, it affects the whole physiology of the organism.
Interesting. So the other thing that we've mentioned too is there's a lot of external cues that regulate circadian rhythms, besides just the light dark cycle. So the presence of food can constrain when animals want to be awake versus asleep, and obviously the food is the source of all the energy of the animal. And what's one thing that's interesting to think about for me is obviously animals can change their diet. Sometimes their diets change seasonally. Experimentally, you can manipulate an animal's diet. We all have firsthand experiences where eating a big meal or eating certain types of food compared to certain other types of food can cause you to get sleepy more than eating other types of food. Do we know about a relationship between food and nutrition and sleep beyond just food availability? Obviously, if you're hungry, it's going to be harder to fall asleep. We've all had that experience. Obviously, if you gorge on a huge meal, you can have a sugar spike and then a sugar crash, and that can affect sleep. But other than those things, can eating certain types of food affect metabolism in ways that affect sleep architecture?
Yeah, I mean, other than what you just described, there have been a number of studies trying to dissect whether certain lipids or protein-rich foods or would affect sleep depth or sleep architecture, and the evidence is not overwhelming. It's again, how we extract nutrients out of the food is it's a slow process, and it's very regulated. And one would be, I would be very surprised if acute changes, or even not, even long-term changes in diet would affect sleep architecture significantly. Sleep architecture being really also extremely tightly regulated. Having said that, there's evidence that, for instance, changes in the microbiome do significantly affect the amount of sleep. And that, of course, microbiome can changes with different diets and so forth, and how our body extracts nutrients from those sources. So there is a relationship between diet and sleep architecture, but it's not easy to dissect out which components of the diet really are critical to affect sleep, because there are a bunch of layers in between, and they're difficult to dissect out.
Mm-hmm. And how about so there's a lot of correlations between sleep deprivation and lots of stuff. I mean, when you sleep deprived animals, lots of things tend to go wrong. But my understanding is there's quite a bit of metabolic dysregulation that correlates, and potentially that's even caused by sleep deprivation. So, for example, obesity rates correlate with sleep quality. The more sleep deprived you are, the more metabolically unhealthy people tend to be. What do we know about sleep deprivation and metabolic dysregulation? Is there much known about the specific ways that sleep deprivation can influence metabolism?
Yeah, at the general level, yes, and sleep deprivation induces insulin resistance, and a whole bunch of especially glucose metabolism is affected by sleep deprivation quite significantly. Actually, in a way that is similar to, and we can talk more about this. Sleep deprivation and aging are two sides of the same coin in many ways. So the body reacts to sleep deprivation in very much the same way as it reacts to aging. So as we age, as we get older, our ability to respond to glycemic challenges is impaired, and same thing as with sleep deprivation. Our the error rate, so to speak, or the accumulation of errors in our metabolic state also accumulates more rapidly as we age, and also with sleep deprivation. So there's really a very good parallel between sleep restriction or not sleeping well, or insomnia, or these conditions and aging. So it doesn't come as a surprise that there are many indicators of aging that are accelerated in people who don't sleep well.
Yeah, well, speaking of aging, I wanted to ask you about sleep across the lifespan. So, again, from experience, we know that sleep obviously changes across the lifespan. Newborn babies sleep all the time, more or less, most of the day. And as we get older, we sleep less. And many of us will know that, our grandparents tend to sleep less in a given day than we do. Sleep quality also often changes as we age. In general, people report that their sleep quality seems to decrease as they get older. Can you talk a little bit about how sleep quality and sleep architecture change throughout the lifespan? And I want to maybe differentiate between developmental changes that are sort of supposed to happen or pre-programmed versus dysregulation that happens due to aging or accumulated damage or something like that.
Yeah, no, that's a great question. So, it's certain that the sleep architecture changes throughout lifespan, and we all know, of course, that infants sleep most of the day. They are essentially growing their brain, myelinating their synapses. There's a whole, really, as you mentioned, a developmental aspect of it that is for which sleep is essential. And as we reach adolescence, then sleep becomes more and more important to either just brain development or memory consolidation as it goes. But of course, at a certain age, yes, there's an accumulation of, as mentioned, metabolic and also genetic errors that result in what I would say, disruption of sleep architecture, mostly due to sleep fragmentation. So it's not that older people sleep more, and they do tend to sleep more, but also it's their sleep becomes disrupted, and therefore, the sense of a recovery from sleep is decreased, and so we published an important paper a couple of years ago indicating that one of the reasons why sleep is fragmented in aged individuals is because there are circuits in the hypothalamus affecting the hypocretin neurons that we're referring to before. So these that hypocretin neurons become more excitable as we age, and that means that the brain, quote unquote, is easier to wake up. And therefore, any either spontaneously or due to external stimuli, the sleep becomes more disrupted and therefore less efficient.
I see. So the orexin neurons, those were the neurons in the lateral hypothalamus we mentioned before. If you're missing those, you have narcolepsy, which is one way to have fragmented sleep. And these neurons promote wakefulness, it sounds like. So if they become more sensitive across a lifespan, essentially the arousal threshold decreases. So it's easier to wake someone up the more sensitive the orexin neurons are.
That's exactly right. That's something that we were able to prove in that paper.
And do we know what drives that? What's actually making them more sensitive?
Yeah, well, in that study, we showed that there's a specific channel. A channel maybe is a protein that allows the flow of ions into the cell, and it's essential for the neuronal excitability. So one of these channels becomes less efficient, and that makes the cell more hyperexcitable, essentially intrinsically more hyperexcitable. What makes that protein less efficient? We don't know, but there are studies indicating that there's oxidation in the external milieu that causes this channel to be a little bit dysfunctional, and therefore affecting the physiology of the hypocretin neuron channel neurons as well.
And so it sounds like there could be the extra excitability that you see emerge in these neurons. The dysregulation of sleep could be related, at least in part, to the accumulation of oxidative damage over the lifespan.
That's correct.
And so, with that in mind, what are some of the things that we know about how to preserve high quality sleep into late life. Are there certain lifestyle factors that might help someone preserve a high quality to their sleep architecture as they age, such as not being constantly sleep deprived, or engaging in other aspects, engaging or not engaging in other types of behaviors that lead to dysregulation of sleep?
Yeah, other than the common sense, unfortunately the basic science has not been able to come up with a protocol that would result in less oxidative stress. It's just that's an intrinsic part of our brain activity, and I again, other than the common sense, lead a healthy lifestyle. I don't think we can recommend anything in addition to that. Yeah.
And so obviously as we age, people often have these sleep problems. One way that people deal with those is through pharmaceuticals. So Ambien and other medications that promote sleep. When we think about sleep medications, medications that are meant to help people fall asleep and stay asleep, to what extent do those drugs actually give you genuine sleep? Are they putting you into a natural sleep state? Are they putting your brain to some other type of quiescent state that doesn't resemble natural sleep? And what are the consequences of that?
Yeah, that's also a fantastic question. So an acute administration of one of these drugs is essentially reduces the latency to sleep. So what these drugs do, most of the sleep aids, what most of the sleep aids do is to increase the overall inhibition in the brain. So those are ligands for inhibitory receptor transmitters. So they reduce the sleep latency. So then the person who takes those drugs has the perception that they slept better because they fell asleep faster. Okay, so one acute administration in principle, and as prescribed by the physician, of course, has not much of a consequence. The problem is when this becomes a chronic administration of the drug. So then the brain tries to adapt to that, to this new state of inhibitory transmission. So and then the sleep architecture becomes affected indeed, and the sleep becomes less natural. And there's also sleep inertia, as many of the patients have experienced. Sleep inertia meaning that they feel sleepy during the morning and after they wake up, and again, and that's part because of the drug itself, and also because of the adaptation to the drug, right? So there's definitely an overprescription of sleep aids in our society for obvious reasons. It's relatively easy to and they're very cheap drugs, and most physicians don't have primary care physicians don't have the training to really go deeper at what caused the insomnia. So they prescribe sleep aids, and that's it's easy, relatively safe as well. So one would say, well, it's a short-term remedy for a problem that may be deeper, and obviously the sleep aids are not the best way to address the problem.
Mm-hmm. I want to ask you a little bit more about REM sleep. So we mentioned it briefly earlier. Obviously, REM sleep is different from waking. It's also different from other phases of sleep, like non-REM sleep. And you mentioned earlier the EEG signature of REM compared to non-REM. So as opposed to non-REM, where you get these large, slow waves of synchronized activity in the cerebral cortex, REM sleep has a very different EEG pattern. Superficially, at least, it almost looks more wake-like than it does like non-REM sleep. And yet, obviously, you're not awake, and so it's not going to be identical to the waking patterns that you see in the EEG. What are some of the neuromodulatory factors that influence the REM EEG? Which neuromodulators are, say, increased or decreased relative to waking in REM sleep?
Yeah, that's an interesting question. So many neurons in the or sleep regulating neurons are active both during wakefulness and REM sleep. So there are very few that are exclusively REM sleep active. So acetylcholine seems to be one of the transmitters that is critical for the non-REM to REM transition. So that is one of the it's one of the basic components of the REM on circuitry, as we call it. But there are others, and dopamine being also one transmitter that is wake on, REM on, and serotonin also. It's at least part of it is REM on. So it's again, there's as I mentioned previously, there's not a single, there's not a simple answer to that. It's just a complex regulation that leads to REM sleep.
I see. So it's complex, but there are some differences between REM and non-REM and waking. So it sounds like acetylcholine is one of the neuromodulators that's quite high activity during REM sleep, as opposed to some of the other ones you mentioned, like noradrenaline, which is very low, little to no activity during REM sleep.
Yeah, that's correct.
And you mentioned earlier too that the theta rhythm that characterizes REM sleep also seems to be important for memory consolidation. So there's some evidence that whatever exactly is going on in terms of brain dynamics during REM, that seems to have some role in memory consolidation, perhaps. One of the things that's weird about REM sleep and dreaming, so dreaming is usually observed during REM sleep, is again, from experience, we all have many examples in our lives where we wake up and we know that we've dreamed, we remember the dream, but then we very quickly don't remember it anymore. It seems to slip right through your fingers. So there's this amnesic quality to dreaming where the experience is very ephemeral and difficult to remember. But simultaneously, it sounds like there's evidence that REM sleep might be important for memory consolidation. So do we understand much about that? Why there's this amnesic quality to vivid dreaming that we experience, and yet there's probably something going on during REM sleep that's important for memory consolidation?
Yeah, I think I would distinguish very clearly what is the dreaming aspect of sleep and the basic function of sleep during REM. So, and we could get into a deeper at what maybe, and this is not really part of my research, but I've been very interested in this topic for a long time. What is dreaming? What is going on during dreaming? Is it an epiphenomenon? It's something that do all animals dream or not, and so forth. So that feeling of not remembering the dream that you just had. It's usually happens when you wake up during a REM sleep, which actually occurs quite frequently, but it's even more so when it's even more obvious when you wake up during deep non-REM sleep. So there is dreaming during non-REM sleep, but there is during non-REM sleep there's no concatenation of experiences of events, and therefore you either don't remember what you were dreaming at all, or you just have a snapshot of the dream and then you forget about it immediately. What characterizes dreaming during REM sleep is this, again, that flow of experiences, ideas that were reactivated or came up again during the dream. So it's interesting that the dreams are essentially an accumulation of the reactivation of experiences, right? We know, for instance, that people who are born blind they don't have they don't have they don't dream images, and most of the dreams are images, right? So they do dream, they do have dreams, but they don't have a visual component to the dream. Okay. So again, it's a reactivation of sensory experiences. Whatever you can sense, you can dream, essentially, right? But we all know that the connection of those dreams to reality is obviously not. It's just not there, and it's likely, again, there are a few studies of brain imaging during dreams, but again, not so solid, I would say. Again, the conclusion of those studies is that essentially you dream what the cortex has lets you dream, or what is available, what kind of the kind of experiences that are available during REM sleep as you dream, and those get filled and connected as if it was a conscious episode, but the fact, of course, it's not connected to reality. It's not. It's just an internal process that gets bound. So and that process of dreaming, again, is very, very different from the physiological events that underneath that are really an essential part of sleep.
Mm-hmm. So we often wake up during REM sleep, but we don't always do that. And I'm wondering what the connection there might be to this concept of sleep inertia that you mentioned before. So very often in my life, for example, I've had the experience where maybe I have a full night's sleep and I wake up naturally 30 minutes before my alarm is supposed to go off. Mm-hmm. And I wake up, I was just dreaming, I feel perfectly awake and rested. But, I've got an extra 30 minutes, so I just sort of lie in bed and think to myself, well, maybe I'll just relax. I don't need to get my day started quite yet. And then sometimes I fall back asleep. And even though I felt perfectly rested before, and now I'm sleeping more, I wake up and then I feel much more tired than I did just a few moments ago. Does that have something to do with which phase of sleep I'm waking up in terms of what dictates the level of wakefulness you experience when you actually come out of sleep?
Yeah, I'm not. I know that. There are a bunch of researchers that study that very aspect of sleep. I'm not very familiar with that literature, to be honest. I don't know exactly what is going on and what are the consequences of waking up at different phases during in the human sleep.
Mm-hmm. One of the things you have been doing more recently, it looks like, is using ultrasound technology to actually manipulate what's going on in the brain. And I want to give people an idea for what's going on here and how this technology is developing. Obviously, when people hear ultrasound, the thing they're probably most likely to think about are the images of the baby in a mother's stomach, which we get from ultrasound. So what is ultrasound, and how is it actually being used by your lab and other labs now to manipulate what's going on in the brain?
Yeah, so indeed, ultrasound is sounds. There are sound waves emitted at frequencies that are beyond our perception, our sensory perception. So there's a very, very broad range of sound is a mechanical wave that transmits through the air, and the ultrasound comprises sounds that from 50 kilohertz or 70 kilohertz to megahertz. So it's a very, very broad range of frequencies. And the imaging, technology uses very high frequencies for imaging, to record essentially those sound waves back and forth the tissue in the soft tissue. So the way we're using ultrasound is slightly different because we're using ultrasound in the brain, and most of the in ultrasound, as in many other waveforms, the higher the frequency, the higher the energy. Okay, and But most of the for the high frequency ultrasound, most of that energy is absorbed by the skull. So for transcranial ultrasound, for neuromodulation, we needed to choose a frequency that gets through the skull and then gets distributed throughout the brain, essentially. So that limits our frequencies, our frequency range to up to 500 to 700 kilohertz. And in turn, the lower the frequency, the lower the energy, and also the broader the focus. So that the focused ultrasound, the transcranial focused ultrasound has this limitation that the size of the focus is relatively large compared to high frequency ultrasound. But we can design tools or tricks to make that focus smaller. But anyway, the reason why we got started into this is because all of the other methods for neuromodulation to essentially target the brain tissue that used magnetic stimulation or electrical stimulation, they don't penetrate deep in the brain. They only affect the surface of the cortex. And I mentioned earlier that the critical players in sleep-wake regulation and in many other behaviors in the brain are subcortical, are structures deep in the brain.
I see. So you needed a way to get down to that depth and manipulate neurons, and most existing ways of manipulating neurons weren't able to get down to the hypothalamus.
That's correct. So the hypothalamus, or even the thalamus, which is also another structure that that is very important in the sleep-wake regulation. So yeah, that was our motivation, our initial motivation. We started more than 5 years ago, trying to figure out ways to use ultrasound and other methods to target deep brain structures. And ultrasound, after consultation with some of our colleagues here at Stanford, we figured, well, ultrasound is probably the method that has not really been fully explored yet, and that there's a lot to be learned from what how ultrasound how neurons respond to ultrasound, and how we can actually manipulate brain activity using ultrasound.
Mm-hmm. And so ultrasound, very high frequency sound waves, so high frequency we can't hear them, but they can penetrate tissue. The frequency that you use with your ultrasound technique is going to dictate how deep the manipulation can penetrate into the brain, and also how focused that energy is. So, you've probably played around a lot with the frequency and what that means for how deep in the brain you can stimulate neurons versus how focused the area of stimulation is. But it sounds like you've actually been able to stimulate neurons and influence neurons using ultrasound all the way down in structures in the hypothalamus. Presumably you guys looked at ones that were going to affect arousal and sleep and wakefulness. Can you talk a little bit about what you found? How specific are these manipulations? Can you get down to very small regions of brain tissue? And, are you exciting neurons? Are you inhibiting neurons? Does that change based on the frequency? What does the manipulation look like?
Yeah, no, that's a great question. So, in short, we can manipulate neurons, we can excite neurons, we can inhibit neurons depending on the frequency of the stimulation. But so our hypothesis, and still a hypothesis because we haven't proved it yet, is that different neurons respond to ultrasound based on the expression of mechanosensitive channels.
So the physical sound waves there's mechanosensors that are evolved to respond to physical stimulation, and that might be what's being tapped into here.
That's correct. So there's people who still argue that this may not be the case, but the fact of the matter is that indeed neurons do express mechanosensitive channels. We don't know exactly why and what for, but ultrasound leverages that the expression of those channels, which is not uniform across the brain. So certain neurons express more channels than others, and therefore are more responsive to ultrasound than others. But again, ultrasound is the response to ultrasound is nonlinear. That means that it's actually very complex. It depends on many factors.
Mm-hmm.
And with our study, we're trying to essentially proof of concept. We're not really trying to get very, very get very lots of details from these, but essentially proof of concept. Do different neurons respond differentially to ultrasound? That was the question we're trying to address.
Mm-hmm.
So in order to do that, Keith Murphy was a postdoc in the lab. He used a measure of calcium activity using an optical fiber that we use all the time, in systems neuroscience, as an objective measure of what was going on in neurons in response to ultrasound. And he showed that different brain regions respond very differently to ultrasound. That if you vary the frequency, you're going to have differences in the response, and also if you vary the repetition of the pulse. So that will also affect how neurons respond to ultrasound. And indeed, they can respond in a positive way, so they can increase their activity, or they can respond in a negative way. That means the ultrasound inhibits their activity. But that's since we had an objective measure of what was going on with ultrasound at individual cell types, now we can build sort of an atlas of how the brain responds to ultrasound in both deep structures and also in the cortex.
Mm-hmm. And so obviously having a way to non-invasively modulate the activity of neurons, even very deeply in the brain, very useful from an experimental standpoint, because now you can excite or inhibit neurons in different regions non-invasively and study circuits in the brain and what the brain is doing. When we think about the applications of this technology to humans, is there are people using it in humans, and what are some of the limitations here? Can you actually stimulate down to the full depth of the human brain? Are people doing that? And could this be useful clinically to, say, treat people with certain psychiatric conditions?
Yeah, of course. That's a very clear application that from ultrasound. So the depth of the of ultrasound is essentially it's unlimited. So ultrasound is actually being used already in the clinic for tumor ablation. At very high frequency, though. Again, as I mentioned, the high frequency, small focus, but high frequency generates a lot of heat, and that's why it is restricted in the clinic to very dose applications, and it needs the patient needs to be cooled. The skull needs to be cooled in order to to avoid other consequences. So
they're using super high frequency ultrasound that's focused on a tumor in the brain. And so someone's got a really bad cancer tumor, you could essentially cook the tumor in their head.
Exactly. Yeah. Wow. And that is being used, has been used in the clinic for a while already. So, again, it's non-invasive, so you don't have to drill, through the skull, but it has its risks because, the heat generated, and of course also the precision of the ultrasound. But anyway, so that's one application. But what we're not talking about that type of ultrasound, high frequency, high energy. We're talking about low intensity focused ultrasound, and but the rule still applies that you can reach pretty much any region of the brain because the sound waves propagate through the brain tissue at essentially the same speed, because for sound purposes the brain is quite uniform, so it's you can get the waves pretty much anywhere at any depth. Mm-hmm. So going back to your question, is it possible to in the future? Will it be possible in the future to treat conditions where neural circuits are not functioning deep? Well, neural circuits deep in the brain are not functioning. I would anticipate that yes, that's possible. I don't know of anyone who is doing it right now, deep in the brain, but it's definitely certainly possible.
So it sounds like we probably don't understand all of the stimulation patterns that might be useful for human applications, but in principle, you can stimulate with ultrasound all the way through the depth of the human brain, and it's non-invasive. So once the technology is developed a bit more and we understand like the consequences of different stimulation protocols and frequencies and things, I would imagine this would naturally be an alternative to something like deep brain stimulation, where perhaps you can stimulate regions deep in the brain without having to cut the person's skull open and implant an electrode deep in the brain.
That's exactly right. Of course, the main caveat now is that, again, the focus is not super precise yet.
Mm-hmm.
But there are ways that one could envisage to narrow the focus by using interference, by using phased arrays, by using a whole bunch of physical tricks, essentially to harness the power of ultrasound and make it more confined to a small volume.
And so, what are some of the ways that you are in your lab using this technology to study sleep-wake circuits in the brain? What are some of the questions you guys are asking?
Yeah, well, one is essentially, can we reach areas of the brain that are complex, like the hypothalamus, where you have a wake-promoting cell right next to a sleep-promoting cell, and are these cells really differentially responsive to ultrasound? So that's one question that we're trying to address. The other is, for sleep and wakefulness and essentially cortical activity. So we know that, there are very complex patterns of activity in the cortex during wakefulness and during sleep at the 100 millisecond time scale. So essentially, there's a lot going on in many different brain modules in the cortex. And with very complex dynamics. So our question with ultrasound would be, if we use ultrasound for a 1 millimeter, so essentially we stimulate a cortical column 1 millimeter wide, how does that affect that complex dynamic of brain activity during wakefulness and during sleep and during REM sleep? So how does the brain adapt to those disturbances questions like that? Yes.
Mm-hmm. And from what I can gather, it sounds like, so when we think about human applications of non-invasive neuromodulation like this, so you have something called transcranial magnetic stimulation, which has been used on people. It's also a pretty coarse-grained way of manipulating brain activity. It's not very focused. Would ultrasound be a more spatially focused or specific way of non-invasively manipulating the brain?
Yeah, and especially the most salient feature of ultrasound is, again, that it can reach deep brain structures which transcranial magnetic stimulation cannot. Mm-hmm. Despite the efforts over the last few years, these deep TMS coils, they do not function well. So yeah, so that would overcome the limitation of TMS.
Interesting. So in terms of what your lab is doing, what are some of the experiments you're working on right now, some of the questions you're investigating in terms of sleep and wakefulness and the underlying circuitry that you're excited about and that you think will have maybe some interesting answers to in the next year or two?
Yeah, well, I mentioned the very complex patterns of activity in the cortex. Those are, I think it's really fascinating. What the new tools are bringing us. New tools in the form of sensors for neuromodulators, and we discussed that. That we only have an idea of single channel recordings of, let's say, norepinephrine, but now with the new sensors we can have simultaneous recordings of multiple transmitters and multiple neuromodulators, and that I think it is really super exciting. Also, the calcium sensors, for genetically encoded voltage, calcium and voltage sensors, they're bringing incredible new images of cortical activity and cortical dynamics that have not been we haven't seen before. So it's trying to decipher what those dynamics mean and what is going on. What are the drivers of those dynamics is also fascinating, and we're really trying very, very hard to get at those questions. What else? So of course we're very interested in the nature of the sleep homeostat. That's I think a fundamental question in sleep research, and we discussed about this a little bit. What is that increases during wakefulness that is released during sleep? So adenosine was a good candidate, but it's certainly not the only one. And so there we have a few candidates that we're trying to evaluate as to their potency for driving sleep. Also, another hypothesis that we're very actively pursuing is whether, as I mentioned, also DNA repair is it a driver for sleep? So we're monitoring DNA repair in real time in cortical neurons as the animals sleep, and see whether we can actually prove that that's true.
Oh, wow. So I would imagine the hypothesis is that some significant amount of DNA repair is happening during sleep.
That's correct. And the reason why is because, as also we discussed with synaptic homeostasis, essentially sleep provides a very interesting window of opportunity to repair DNA that has been damaged during wakefulness. And why do we need sleep is because, of course, the DNA repair also happens during wakefulness, but the rate of damage is much less because there's less activity in general, and that allows you to essentially to catch up with the DNA repair rate. And also we are seeing that not only that, but also during sleep there's a higher rate of the DNA repair. So that would also rebalance. That. And needless to say that DNA integrity is a fundamental property of nervous systems, and that would explain why, even with very, very different brain architectures, we see sleep across in all animal phyla. So again, we believe that sleep has really to subserve a really, really fundamental function, and one of these could be DNA repair.
Interesting. We covered a lot already. This has been fascinating. Is there anything that you want to reiterate for people that we went over, or any final thoughts that you want to leave people with regarding sleep?
Well, I just want to restate that sleep is universal, and it is universal for a reason. I think we need to take care of our sleep very well. I think the consequences of not sleeping are really very, they're very detrimental to our health. So we cannot ignore the fact that we're not sleeping well. I would really, for those who have trouble sleeping, I would really, really encourage them to get the help of a physician and hopefully get referred to a sleep clinic, because it's it's something that can be fixed in the hands of professionals now, but it cannot be ignored. So that will be my final point. Yeah.
All right. Well, Professor Luis de Lecea, thank you for your time. This was fascinating.
Thank you so much. I really enjoyed this.
Supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.